Good morning, students. Now coming to pollen pistil interaction. Okay, already know outbreeding lyrics in pollination. Now coming to pollen pistil interaction. Students, pollination is not a guarantee in cross pollinating plants because it requires the transfer of pollen from one plant to the next plant. So, cross pollination is not a guarantee. That is, the transfer of right type of pollen grain to the stigma of gynoecium. It's not a guarantee because sometimes stigma it may receive. Okay, stigma may receive either the pollen grain of the same species, at the same time may receive the pollen grains of another species. The pollen grain of the same species, this is considered as compatible strain. Compatible. This is called compatible pollen grain. This is compatible pollen grain. But at the same time, pollen may receive the pollen grains of stigma it receive the pollen grain of the same species, means compatible pollen grain. At the same time, another species, pollen grain of another species, these are considered as incompatible. They are called incompatible pollen grains. Okay, these compatible pollen grains are called right type of pollen grains, and incompatible pollen grains are called wrong type, right type, and wrong type of pollen grains. When the stigma receives the pollen grains of compatible strains and the pollen grains of incompatible strains, now the stigma it has the ability to recognize whether the collected pollen grain is right type pollen grain or wrong type pollen grain. Stigma has the ability to recognize whether the collected pollen grain is right type pollen grain or wrong type pollen grain. So, if it recognizes the right type of pollen grain, it allow the pollen grain, okay, it allow the pollen grain for post fertilization changes that lead to fertilization. So when stigma receives the pollen grains of the same species, it allow the pollen grain and it accept the pollen grain, it allow the pollen grain to post fertilization events that lead to fertilization. But when the stigma receives the wrong type of pollen grain, okay, sometimes stigma may receive the pollen grains of another species, incompatible species, means wrong species. Now stigma it reject the pollen grain. It reject the pollen grain. How it reject the pollen grain? By inhibiting the germination. Okay? Or by inhibiting the growth of pollen tube. So, stigma has the ability to recognize whether the collected pollen grain is right type or wrong type. When it receives the right type of pollen grain, means compatible pollen grain, it accepts the pollen grain and it allows the pollen grain to post fertilization events that lead to fertilization. But when it recognizes the pollen grain is wrong type, now it rejects the pollen grain and it rejects the pollen grain by inhibiting the germination of the pollen grain on the stigma, or the growth of pollen tube through the style. So, this acception and interaction of the pollen grains, acception and resection of the pollen grain by the stigma, it is a continuous dialogue. This is a continuous dialogue that is mediated by the chemicals. Okay, this continuous dialogue between stigma and the pollen grain, it is a continuous dialogue. It is mediated by chemical substances which are secreted by pollen grain and those interact with the stigma. Now this type of chemicals, they are also isolated by scientists. They are also isolated by the scientists. So this is pollen grain. Maybe this stigma, it has the ability to recognize the pollen grain. It is right type or wrong type pollen grains. Similarly, very important. When the stigma, when pollen grain is fall on the surface of stigma, it germinates. It germinates on the stigma. Now it produces a small pollen tube. A small pollen tube. For example, this is the pollen grain, and this pollen grain, it germinates on the stigma and it produces a small tube-like structure. This is called pollen tube. Now this pollen tube, it is entered into the style. Finally, the pollen grain, it is entered into the ovule. In 60% of angiosperms, in most of the angiosperms, pollen grains, they are released from anther at 2 cell stage. At this situation, this one is vegetative cell and second one is generative cell. So, in 60% of plants, pollen grains are released from anther at 2 cell stage. One is vegetative cell and other one is generative cell. So these vegetative and generative cells, they enter into the pollen tube after the germination of the pollen grain. But in case of remaining 40% of plants, these pollen grains are released from the anther at 3 cell stage. One is vegetative cell and remaining 2 male gametes. 2 male gametes. So, in 40% of plants, pollen grains are released at 3 cell stage. One is vegetative cell and remaining 2. They are 2 male gametes. So, in case of this 40% of plants, this pollen tube, it carries 2 male gametes from the beginning. It carries 2 male gametes from the beginning. But in case of this 60% of plants, first vegetative cell and generative cell, they are entered into the pollen tube. While the pollen tube grows through the style, that is inside the stigma, this generative cell divides by one more metaphase, leading to the formation of 2 male gametes. Students, here this pollen tube, it carries vegetative cell and generative cell in case of 60% of plants. Then this generative cell divides by one more metaphase to form 2 male gametes. But in 40% of plants, already 3 cells are present inside this germinating pollen grain, so that this pollen tube carries vegetative cell and 2 male gametes. It carries the 2 male gametes at the beginning, that is along with the vegetative cell. Here, once the pollen tube, it reaches to the ovary. First it reaches to ovary and then it is enter into ovule, then it enter into ovule. So the entire pollen tube into the ovule, it is of 3 types. One is porogamy. One is porogamy. Okay, porogamy. For example, this is ovary, and this is ovule. This is ovule. Okay, this is ovule. Here, this pollen tube. This is pollen tube. It is directly enter into the ovule through microfile. The entry of pollen tube into the ovule through microfile. This is considered as this is called porogamy. This is porogamy. Best examples for this porogamy. This is common type of entry of pollen tube. This is common. Example, Botany is one of the example. But in some members, the second way of pollen tube into the ovule. This is called chelazogamy. Chelazogamy. Here, for example, this is ovule. Okay, and this is ovule. Here, pollen tube. It enters into the ovule directly through chalaza. Pollen tube it enters into the ovule through chalaza. That is called chelazogamy. It is discovered by scientist True. Discovered by True in Casuarina plant. Casuarina. Discovered by True in Casuarina. At the same time, there is another way of entry of pollen tube into the ovule. This is called mesogamy. This is mesogamy. For example, this is ovule. Okay, this is ovule. Here, pollen tube it is directly enter into the ovule by observing through the integumentary. Sometimes it may be enter into the ovule through the funiculus and integumentary. So, the entry of pollen tube into the ovule through integuments or funiculus and integumentary. This is considered as this is mesogamy. This is called mesogamy. Example, this is best example, cucumber or cucurbits, cucurbita or cucumber. Members belongs to family Cucurbitaceae. So this is porogamy. Ovule in the pollen tube enter into the ovule through microfile. Example A. This is second one. This is chelazogamy. Discovered by True in Casuarina. Here pollen tube enter into the ovule through chalaza. And this is mesogamy. Example, cucurbita. Here, this pollen tube will enter into the ovule through integuments or integuments and the funiculus. That is the entry of pollen tube. Similarly, all the events in sexual reproduction, okay, from the deposition of pollen grains on the stigma until the pollen tube is entered into the ovule. All the events of sexual reproduction from deposition of pollen grains on the stigma to entry of pollen tube into the ovule, and all this collectively they are called pollen pistil interactions. Pollen pistil interactions. Students, now coming to other points. Artificial, okay, artificial cross pollination. Artificial cross pollination. So in artificial cross pollination, you know, plant breeding program. There are some events in plant breeding program conducted by any plant breeder. One is emasculation. One is emasculation. And second one is bagging. Bagging. And third one is artificial pollination. And final step, this is rebagging. Rebagging. So these events, they are necessary in plant breeding program. These events are necessary in plant breeding program. Okay? Emasculation means the elimination of anthers or the removal of anthers from the bisexual flower of female parent in a plant breeding. That is considered as emasculation. The removal of anthers from the floral buds while the flowers are present in a bud condition, that is considered as emasculation. This emasculation prevents self-pollination. Then bagging. Emasculated flowers, they must be enclosed by polythene bags or butter paper bags. Why they must be enclosed by polythene bags or butter paper, suitable size butter paper, to avoid pollination by unwanted pollinates. So bagging it away, bagging and hybridization it away contamination by foreign pollinates. Unwanted cross pollination is completely avoided by process of bagging. Similarly, after the attaining of stigma to receive the pollen grains are collected from male plant and are dusted on the stigma of the female plant. This is considered as artificial pollination. Similarly, rebagging. After the completion of artificial pollination, again these flowers must be enclosed by the same butter paper bags. That is called rebagging. Students, very very important. Arrange the correct sequence of these events in plant breeding. 1. Emasculation. Okay. 2. Bagging. 3. Artificial pollination. 4. And final one, this is rebagging. Similarly, students, double fertilization. Double fertilization. Okay. So double fertilization is the characteristic feature of angiosperms. It takes place only in angiosperms. Okay. Here one male gamete. First male gamete. In angiosperms, the first male gamete. Okay. It is fused with the female gamete, leading to the formation of zygote. Okay. The fusion of the first male gamete with the female gamete egg, that is considered as fertilization. This is called fertilization, and this is also called syngamy. Is also called syngamy. As a result of syngamy in angiosperms, it leads to the formation of zygote. Male gamete is haploid. Female gamete is also haploid, and resulted zygote is diploid. So, fusion of first male gamete with the egg cell. It is called fertilization or syngamy. It was discovered by Strasburger. Okay. At the same time, second male gamete it is fused with the secondary nucleus, which is present in female gametophyte, that is in endosperm. It leads to the formation of primary endosperm nucleus. This is haploid and this is triploid. Resulted primary endosperm nucleus is called, it is triploid. Fusion of second male gamete with the secondary nucleus. This is called triple fusion. This is called triple fusion. Why this is considered as triple fusion? Total 3 haploid nuclei, they are involved in this triple fusion. What are the names of this haploid nuclei? One is second male gamete and remaining 2 polar nuclei. Already 2 polar nuclei, they are fused to form secondary nucleus just before fertilization. Now the second male gamete is fused with the secondary nucleus, leading to the formation of a primary endosperm nucleus. Here both syngamy, okay, and triple fusion. Singamy and triple fusion both are considered as double fertilization. Double fertilization. Okay, so in angiosperms, one male gamete is participated in singamy. Second male gamete is participated in triple fusion. Such participation of two male gametes in two different fusions in angiosperms, that is considered as double fertilization. So, as a result of double fertilization, primary endosperm nucleus is formed. This primary endosperm nucleus is the initial cell for the development of endosperm. Okay.